with the last webinar of the ONTOX-VHP4Safety webinar series. Today we have 4 presentations, one by Trine, one by Susana, one by Julen, and one by Fabian. The first 3 are representatives of ONTOX, and Fabian is representative of VHP4Safety. We will start, as always, with telling a bit about the projects. Yes. So VHP4Safety is a Dutch research project. It's funded by the Dutch Research Agenda, the NGRA, which is a program of the Dutch Research Council, the NWO. It's coordinated by 3 organizations: Utrecht University of Applied Sciences, or Hogeschool Utrecht in Dutch, and RIVM. And it runs from June 2021 until May 2026. Then we go to ONTOX, which is a European research project focusing on similar goals. Both projects focus on NEMS, and it receives funding from the European Union's Horizon 2020 Research and Innovation Program. It's coordinated by the Vrije Universiteit Brussel, and it runs from May 2021 until April 2026. And well, as I just said, these are 2 projects with similar goals, one on a Dutch level and one on European level. And because these projects both have the common goal of advancing human risk assessment without or reducing the use of animals, they set up a collaboration. And well, this is one of the babies of this collaboration, this webinar series. The first meeting between the 2 projects was held in October 2022, and then 6 collaboration groups were formed in 2023, and a memorandum of understanding was signed in February 2024. So these are the 6 collaboration groups. So these are the areas in which we collaborate. Actually 5 of these collaboration groups have also presented a webinar. So the collaboration groups are research output management, previously called data management, stakeholder involvement, QAOP, chemical selection, IT platform, and risk assessment. And today we are present at the webinar of this one on the bottom, the collaboration group risk assessment. So this webinar series, as I already said, this is the last one of the series. Each webinar is focused on one collaboration group. We consider this a nice opportunity for early stage researchers to present their work. We had these webinars approximately once every 2 months, and the recordings can be found on YouTube. And as soon as the recording of this one becomes available, it will also be shared on the LinkedIn pages of ONTOX and VHP4Safety. Then I give the floor to Marie to tell a bit about ASPIS Academy.
Yeah, so this webinar is organized jointly by VHP4Safety, which is represented by Nick, and ASPIS Academy, of which I am one of the representatives. And ASPIS Academy is a real network for early-stage researchers involved in new approach methodologies in toxicology, and we're trying to connect researchers, boosting their careers by giving them training and essentially trying to promote the work of early career researchers. If you can put the next slide, Anik. So in particular, we have one of the prongs of the ASPIS Academy is a training program. So we are organizing in-person trainings, for example, at ASPIS Open Symposia and at project meetings. We organized a summer school, for example, back in April in Valencia all about data. And we organize online webinars and workshops, so both about soft skills, such as science communication, funding, effective presentations, but also hard skills. For example, we organized a couple of years ago, AI in risk assessment, cell-based test method development, this kind of things. And the last slide about the ASPIS Academy is about the people. So we are a group of researchers who are trying to organize these. Julen, who is going to present today, is actually part of this. He's actually the chair of ASPIS Academy. And there's a few more people from each ASPIS project that are involved. And we also have the board with coordinators from each project. And if you want to join us, you have the QR code here. You can join the LinkedIn group to stay in touch, stay updated about the activities that we organize, and you can reach us via the email address that is at the bottom. And now time for the actual meat of the webinar. So Nick, go for it.
Yes. So after this introduction, we go to the first presentation by Trine. Trine, can I ask you to share your slides?
Then hope for success, huh?
Yeah. Yes.
Maybe one last thing before we get started, that Trine actually needs to leave after her presentation. Yes. So if you have any question for her, make sure to put it in the Q&A during the presentation so we can ask them immediately after. That's actually valid for every presentation. Please put your questions along the presentation in the Q&A. We'll monitor it and then we'll ask them at the end of each presentation. There will be a short Q&A after each.
Yes, thanks.
Okay, now I cannot see you anymore, but hopefully that you can see my presentation.
Not yet.
Not yet. Then that was very strange because I haven't done any. I did share it and then I think it's okay. Let's try again.
Yes, now we see it. Not in presentation mode yet.
And if I just want to reduce the
Yes, perfect.
screen.
Perfect.
Yeah, that's good. 2 attempts, it's not that bad, huh? Okay, so I'm going to talk about risk assessment of chemicals using NAMs and also specifically the case study of P4 that we work on in ONTOX. And I first want to say a bit about risk assessment. I'm not sure how many risk assessors that will listen to this. So for those that will be very familiar, but for those who don't do risk assessment, it might be good to hear something about risk assessment in general. And I'm going to start very basic about hazard. So hazard, this is a typical hazard. You can feel that this is inherited hazard meeting this animal. But you can also have a hazard with no exposure, which means no risk. And also, I think then if you do in vitro studies that we would like to use in some of these risk assessment, that is hazard. You need also then to have an exposure to have a risk, which we have here. And it's also for risk assessor, it can be sometimes not always clear the definition between hazard and risks. That's why we have to stress this and do it very clear every time we talk about risk. Is there an exposure or not? And I'm going through this very familiar process as well, so that you can see the whole process of risk assessment of chemicals. We start with hazard identification. That means for a chemical, do it have any type of harm based on the chemical structure and inherited ability of a chemical to make a hazard. That's the first thing we do, and it can have several hazards. Then the next step is to characterize that hazard, to make a dose response. What kind of dose will this hazard occur? And most often, the hazard will be using studies from animal studies or human studies. Very few in vitro studies are used today in practice when it comes to risk assessment. I have my experience mostly from the EFSA as an expert there, and I know that we have used GeneTox is used regularly for hazard characterization, but in many other areas we don't use in vitro studies, and mostly it will be animal studies and human studies. So then we will then do the hazard characterization based on animal studies and human studies. In addition to that, you have to assess the exposure to humans. What kind of route of exposure do we have? What is the amount in the product or in the food, the frequency of use or intake, and the time of exposure. And first, when you combine these two, hazard characterization and exposure, you will do the risk assessment. And then we talk about risk to human health. I mean, environmental risk assessment will have different processes, and often they are based on hazard identification solely. So this is human risk assessment. And then risk management is not done by the risk assessor normally. That's done by the commission. And risk communication can be done both by risk assessors and risk managers. So this is the process of risk assessment. I will now go over to the P4 case study, and I will start just to talk a bit about P4 in general. Many of you know the chemical well. Maybe some of you don't know it so well. And when it comes to the exposure of P4, we do that for humans often with two different methods. You can either estimate it by measuring concentration in products or in food. And then in human studies, you have assessed the frequency of use or intake. This is a frequency questionnaire from the mother and child cohort in Norway, where you have asked people how often they eat different kind of food. But of course, you can also ask whether they use cosmetics or how other questions that you can also combine with the concentration in products. And then you can estimate what kind of exposure you have, the external exposure of these individuals. Another way of doing exposure estimates are to measure directly the concentration in blood and urine from these individuals. And we have many times we have both possibility to do the exposure estimates based on frequency questionnaire or diaries, or and measure it in blood and urine, and you can compare them. When it comes to the concentration in blood and urine, that will be a very good measurement of the total exposure from all kinds of sources. So how can we be exposed to PFAS? For PFAS specifically, we can be exposed through inhalation, indoor air or outdoor air, skin contact, through dust or different kind of products, cosmetics among those. And we can have oral intake from food and drinks, and also dust, which has been also calculated for some studies. So, but the major source of P4 and PFASs are diet, by far. And this is from a small study in Norway called ATIP, where they have estimated the intake from different sources. And you can see the diet is really a major source and that's also the same for P4. This is from a study of Euromix. Here you can see that the figure on the left-hand side, we have calculated the exposure both from diet and from cosmetics. And each of these dots are one individual. And you can see that some of these dots overlap with diet exposure. And that means for some individuals in this study, it was as high exposure as from diet, from the cosmetics. But still, the major source for most of the individuals were from diet. When it comes to biomonitoring, measuring the different PFASs in blood, blood is by far the best biological way to measure the PFASs, at least the long chain PFASs. And you can see that P4, you have 4 different PFASs here, and the sum of 4 PFASs. And P4 is the second most high, with the highest concentration in blood. PFOS is normally higher concentrations in blood compared to P4. But then, of course, when you do the biomonitoring, you don't know which sources the concentration come from. That's only measurement of the total exposure. And we know quite some we have quite some information on the toxicity of the P4. This is taken from the EFSA opinion from 2020, where they assessed the risk assessment of several PFASs, among them PFOA. And we know that PFOA have an effect on the immune system. Actually, the TDI is based on the effects in the immune system of humans, where they saw suppressed antibody development after vaccination. And that means you can have an increased risk of infections. It's also shown that PFOA have effects on the fetal development and also effects on liver. Increases liver weight in experimental animals, and this is also at human relevant doses. And you also saw that when it comes to the exposure, nearly 30% of children and adolescents have higher blood levels of PFAS than the acceptable levels. So this means it is a risk being exposed to PFOA. And then, of course, you can ask, why do we want to do another risk assessment of PFOA when we have already quite some information? The reason why we selected PFOA is because when we use in vitro studies, we would like to see, it's good to try that out with a substance that have some information already. Then it's easier to compare it to the already established risk assessment. In addition to that, in ONTOX project, it was most of the partners could contribute to the risk assessment if we selected the PFOA as a chemical, because they have tested PFOA in different in vitro studies. And also that's why we selected PFOA in this case. So here is the OPERA framework. This has been talked about a lot. This is a framework that has been developed in the ONTOX project. You can recognize some of the elements from the general risk assessment scheme. We have exposure in the green here on top, and we have included TTC. We have biokinetics included. We also have the hazard here, and you can see that some of the elements are the same, but in this case we would like to introduce new methods in these boxes compared to what is normally used in a risk assessment. If you do a risk assessment in EFSA, you will have a traditional way of doing that, but we would like to introduce new methods. We would like to introduce AI tools to do this, and we would also like to try to do this risk assessment in a probabilistic way, which means that we would do the exposure assessment in a probabilistic way, and also the hazard will be done in a probabilistic way, and we will use in vitro studies. And that's why it is good to try to do this for a chemical that you have quite some information on already. Yeah, this is more or less the same that I informed you on already. Exposure distribution, probability, biokinetics, probability of hazard, and the potency estimates from in vitro and from in silico, I forgot that. And then the probability of risk for individuals and the population in the end. So to stress the importance of exposure, it has it's important to also be aware that exposure assessments are really important to do a full risk assessment. In this case study, we will do focus on the oral exposure, and we will do probabilistic external exposure estimates from the EuroMix study that I already presented some data from. That will be individual external exposure. And then we have also done a population exposure estimate from using the EFSA exposure data that was presented in the opinion from 2020. There you have some summary data. I think you have P50 or the mean and the P95 for several countries. And what we have done then is to use that and then model the whole distribution from these 2 points. And you can see an example of that on this figure. This is for children. We have modeled the whole distribution, and this is a cumulative probability plot. and you can see that it's quite some children that will exceed the TDI for some of the countries. This is external exposure from the diet. And then to translate that external exposure into internal exposure so that we can compare it to results from in vitro studies, we will use the PBK modeling to get the internal exposure. And Susana will talk more about the PBK modeling approach in the next presentation. And then, of course, you can have an internal exposure in plasma, you can have it in liver, and you can have it in kidney and organs that you are interested in for the toxicity of the chemical that you are interested in. In this case, it will be liver and kidney and plasma probably. So just a bit, very short about the application of PBK modeling. It has been used in risk assessment for some time already. Here you can see that it's a kind of figure on the right-hand side describing very schematic how PBK model can be done. You can have different compartments that you decide is important for your chemical. In this case, you have lung, fat, muscle, skin, and liver. And each of these compartments will have a mathematical equation then, a dynamic equation. And it has been used in risk assessment already in BPA risk assessment in 2015. We used PBK modeling to extrapolate effects between animals and humans. In this case, we would like to use the PBK modeling to estimate the internal exposure in humans. And also we would like to have a dose, know the P4 doses at the target organ, liver and kidney. And when I first had a PBK course many years ago, the teacher of that course said to me that PBK models never reflects the full truth, but they can be very useful. And that's the truth, that this is models with a certain uncertainty around it, and it will never be exactly the truth that you will get when it comes to the modeled exposure. But it gives us an opportunity to actually estimate the internal dose and compare it to the in vitro studies that we do in ONTOX. So then to hazard characterization. Then we do often base that on animal studies or human studies, not very often on in vitro studies. The outcome from the characterization is a point estimate. And you can see in the figure here that you have a study that you have tested some doses of chemical, probably. And then you have fitted done a curve fitting to those data points that you have produced from a study. And then you can decide on the benchmark response. That's the dose that gives a non-adverse increase in effect. And that's a toxicological judgment. And you need experience from your studies to decide on what is a reasonable BMR. In this case, they have selected 5%. And then you decide on the dose that gives a 5% increase in the effect. And then you look at the variability in the data that will be presented in the modeling. And then you estimate the benchmark dose lower confidence interval. And that will be the point estimate if you use a benchmark modeling. Otherwise, you can also say that the lowest dose that don't give an effect will be the no adverse effect levels. This is the normal way to do it. But if you're going to do a probabilistic hazard characterization, you would like to have the whole distribution of possible BMDs. And that's something that is not very often used. It's quite a new method. And we struggle quite hard to try to figure out how to do this. And some of the tools available, you don't get that information. So that's a bit of the problem. Recently, just a second. We discovered that the new web tool presented by EFSA in 2024, they actually present the whole distribution of BMDs. And And then, but they don't read, the web tool don't give you the numbers. But we also saw that they presented our package that is the basis that you can use for the modeling, and then you can also then extract the whole distribution of the BMDs. And in the figure here, you can actually see that this is in vitro data of kidney after 24 hours of P4 exposure produced in ONTOX. And I have used our package BMA BMDR from EFSA. You will get the same figure if you use the web tool, a BMD calculation. They produce exactly the same type of figures. And then you can see that this orange distribution is the distribution of the BMDs. In this web tool or in our package, they don't select one of the models to fit the data. They do something called model averaging. And that means that they have several models that will be run to see whether they fit the data that you have produced. They will weigh the models differently according to how well they fit the data points that you have produced. And then they will use Bayesian statistics to do a model averaging. All this is done in the web tool as well. In the R package, they do it as well, but then you have to, I had to struggle a bit to get out the distribution, the numbers, but we managed to do that in the end. And then you can, of course, present these data the way you would like it if you use the R package. And then these data that we have extracted from using the R package of the benchmark doses, we have done it for different, the left-hand side, you can see that the benchmark response 10, 20, and 30 have been done arbitrarily just to see how it looks. And we have then taken out the BMD distribution. And then the model averaging using the bridge sampling. You have two different ways of doing the model averaging in this R package. You also have that in the web tool, by the way, but it's only the bridge sampling that give you all the numbers. And then we take the numbers out and made new figures. And then you can see on the left-hand, on the right-hand side here, you have the cumulative probability. And then you can say something about the probability of the hazard. But of course, what to select when it comes to the benchmark response, that's again a toxicological judgment. And it's not given. I would say that benchmark response 30 seems to be maybe too high, but this have to be discussed with the people that know something about kidney toxicity to decide what are reasonable benchmark response. In addition to this, we will in this case study also use physiological maps. That means we want to translate toxicology into biology. This is a slide from Bernard, which in ONTOX developed the physiological maps. That means you go from chemistry, using kinetics and use information on toxicology and translate that into biology. And this is outside my expertise, but we will use it in a case study to try to translate the in vitro liver effect into a probability of liver disease using Boolean models. And this is something that we have not done yet. This is work in progress. But this is something that will be included in the probabilistic risk assessment of P4 in the end. So to the end, probabilistic risk assessment, we need a probability of exposure, not only point estimate, we need a probability of hazard, which is more complicated than the probability of exposure. It's not done very frequently, while the probability of exposure has been included in some risk assessment already. And then you have to combine them. And as you saw, I have no slide on the probability of risk yet, because that's also work in progress. We have not finished doing the probability of hazard, only tried to very beginning of using the method. And then we will compare the probability of exposure and the probability of hazard, and see what we will end up with when it comes to probability of risk using the in vitro studies in ONTOX. We also have done a literature research, so we will also extract some information on P4 hazard from literature, and then hopefully we can combine the in vitro data that we have produced in ONTOX together with data that we have from literature. So this is how far I have come with the case study on P4, and there's a lot of collaborator across the ONTOX. Most of the partners are involved in this. So then I stop sharing.
Yes, thank you for this very nice overview, Trine. If people have any questions, they can post them in the Q&A. Right now, there is one question, which is the OPRA framework published?
No, it's not published yet. I think they plan a publication, but it's not. I think it's also something that they work on. So
Okay.
that's something that will be published as a part of the ONTOX project.
Yeah.
Mm-hmm.
Oh, another question. What was the rationale for selecting this specific in vitro model, and how well is the model validated?
In vitro model of which model do I think? I think Julen will talk about the liver.
Yeah, I think the in vitro model will be discussed in the third presentation. Indeed.
Yes. So then I guess Julen can argue for his models, but I think P4 is known to cause liver toxicity. That's one of the reasons for selecting liver. Kidney is an important organ for P4 as well. It's reabsorption in kidney, and it's also some kidney toxicity. So I think that's maybe the reasoning why we use this also for P4. But P4 is not the only chemical that is studied in ONTOX. This is just selected for this case study. It's a lot of chemicals that has been studied in ONTOX.
Yeah. Juliet clarifies that she indeed meant the kidney model.
The kidney model. Okay, that's probably, I mean, I don't know that you have to select something when you do this. And I don't know exactly why the reasoning for selecting the kidney. When it comes to the P4, we used, I mean, for the ONTOX, you have selected to focus on kidney, liver, and brain as the organs to select. And then, of course, you can say that for the P4, kidney and liver is already known to be important. But in ONTOX as general, you study a lot of other chemicals as well. So that's
yeah. I hope that answers her question. Then I think if there are no more questions, we would like to thank you very much, Trine. I think you have to leave, right?
Yeah, I have to leave. Sorry for that.
Yeah. Then we go to the next presentation, which is by Susana.
I will share.
Perfect.
Hopefully you can see it. If not, just tell me if you can't.
Amazing.
So indeed, I mean, it was actually very nice that Trine presented before, because I think she will already make this presentation even a bit clearer. Because indeed, I'm going to introduce the PBK and the QVIV, and a bit specifically what we're doing for the case study of P4. So a bit first, because not everyone knows kinetics very well and why we need it. So obviously, Trine already presented a bit of this risk assessment framework, but what I want to emphasize nowadays, when you do this risk assessment, you usually use animals maybe to do hazard characterization, and then we actually want to evaluate human exposure. So there's quite a bit of extrapolation here between species, but also between the population and individuals. And typically there are empirical uncertainty factors that are taken into account. So there's usually uncertainty factor of 10 between species and an uncertainty factor of 10 between for the human population, for example. But these, because they're empirical factors, they don't really represent mechanistically for the specific chemical. So ideally, in this next generation risk assessment framework, we want to have chemical specific adjustment factors, hopefully even avoiding completely animals, especially if we can do the hazard characterization in vitro, then we still need to then evaluate, what are the uncertainty of performing this extrapolation. And so a bit for the kinetics, so this is really linking your internal concentrations to the external exposure. So for that, we need to understand the ADME processes, so absorption, distribution, metabolism, and excretion, because you can have different absorption routes. As Trine said, you can have dermal, inhalation, and oral. There's even other exposure routes, like, for example, ocular. And then your compound, once it gets absorbed and then is in your systemic circulation, you can distribute into tissues. And obviously, it's important to understand that for both toxicological, but also for the pharmaceutical sciences, it's usually assumed that only the unbound compound can actually interact with the target. So if a compound has heavy distribution into tissues, it might not be available to actually cause its action, for example. And then you have your metabolism excretion. So this is how a compound might disappear from the system. The main organs there, for example, you have your hepatic metabolism or biliary excretion, or you can have kidney excretion, which we'll see is very important for PFOA. So typically, these kinetics, or originally they were incorporated or they were modeled through compartmental models, which are these semi-empirical models that just try to recapitulate the kinetics that we see either in animal or in the humans. But obviously, because they are not fully mechanistic, it's a bit difficult to perform extrapolations and to actually use the mechanistic knowledge we know from our organisms. So there's where physiologically based kinetic models come from. We actually use the physiological knowledge we know from our body and try to make a bit more mechanistic predictions. So in this way, they distinguish from compartmental models in that actually the compartments are the organs or the subcompartments of the organs, for example, your cells, your endothelium, your plasma. So what do we need to parameterize PBK models? Obviously, we need the physiology that I just said, and this physiology depends, if you're incorporating a human or a rat, or if, for example, we want to parameterize a pregnant woman. So we can already adjust to the physiology we know. It depends obviously on the chemical specific properties, and this can be, for example, your lipophilicity, but there's also properties that depend both on the type of individual, but also on the chemical. And these are, for example, the metabolism. It depends obviously on the chemical, but it depends on the individual. If it has polymorphisms, if it's, again, a pediatric, or if it's an elder person, the metabolism might be different. So there's quite a few parameters that actually play on both the chemical and individual, and maybe more sometimes on the pharmaceuticals, but not only. Sometimes you also need to understand, how is the chemical formulated, or, for example, how do we ingest it? Is it with food, or is it in an empty stomach? How often do we assume the person is taking this compound, and also, for example, if the person is doing exercise. And so if we have all these parameters, then we want to solve our PBK model and perform simulations. And so for that, we need the software. And obviously, because PBK models basically use the systems of differential equations, any system software that can solve differential equations can actually be used. Theoretically, Excel can be used. Is it the best software? Probably not, because it's going to be very heavy, but it is possible. And obviously, one of the most common softwares nowadays is to use R, where people will develop the PBK model from scratch. And it's very good if you want to really tailor to specific aspects, but it is very laborious, especially if you have to start from zero, from scratch, because you have to build all the model. There's a lot of errors that can occur when you build your model. You need a lot of physiological parameters, as I indicated. So you also need to do a huge collection of the literature. And it's also very important for any in silico tool to really also ensure the quality control. So obviously, if it's simulating what you think is simulating, if there's no errors, but also to maintain the sustainability so that the model that I create now in 5 years is still working, even though the software is updated. So obviously, to build sometimes PBK models without proper softwares might be difficult for regulatory or industry needs, for example. So this is why there's quite a few commercial softwares. You have SimSip, you have GastroPlus, for example, then you have the system that are used in ONTOX, which is the Open Systems Pharmacology Suite tools. And so this is open source software where you can develop PBK models. It already kind of contains a pre-made PBK model where then basically we can change some of the aspects. And there's already the database of physiologic parameters for the different populations. There's also free training for academic researchers. And there's also a library of qualified models for different molecules, mostly pharmaceuticals. Although we can develop the model using the software, which has a proper interface, we can then also migrate it to R if you want to do more specific things, which is the case for P4. And to say, like, where do PBK models sit in this next generation risk assessment? Because PBK models are not intrinsically an M. They were a model already used for a few years, and typically they will even be actually parameterized based on animal data. For example, they would fit maybe the metabolism and the absorption to animal data. But nowadays, as we get more in vitro data, so the idea is that we can also parameterize the PBK models based on this in vitro or in silico data, and to make it as mechanistic as possible. Then we can also avoid species extrapolation. A few examples, for example, for absorption, you can use CACO2 assays. For distribution, we also use some in silico methods to make predictions. And for metabolism, we can use hepatocytes. And so we have quite a few ways of parameterizing the models of the. Obviously, there's a lot of in vitro systems, and the challenge nowadays, there's still no OECD test guidelines approved for ADME assay in vitro assays. And one of the challenges, obviously, we also need what you test in the lab is not exactly what the equivalent, for example, to your liver. So you need some scaling factors, and all of these needs always, for each in vitro method, we need really to understand how this IVIV needs to be performed to have accurate predictions. And so the other aspect in the next generation risk assessment is exactly how PBK model can actually help using in vitro effect concentrations in the risk assessment, because the effect concentration you test in your in vitro system, it's not exactly those in vivo, neither is necessarily equivalent to the concentration in plasma, actually. It would be more equivalent to the unbound concentration in their specific tissue. So for this, what we need to do, as Trine said, so if you have your dose response curve, and we usually suggest also to really trying to understand where is the unbound concentration in this in vitro system. You do your benchmark. Hopefully you get it actually with also the interval of confidence. And with this, we can perform reverse dosimetry, meaning, so if I assume this is equivalent to, for example, unbound concentration in tissue or unbound in plasma, then I can fit actually what will be the external dose that would lead to this concentration internally. And this is what we call then reverse dosimetry. And so what you get in the end is, for example, a human equivalent dose. We have more experience with oral equivalent dose, but obviously there's also for other exposure routes. And then these you can actually compare to known exposure doses. The challenge that we have for P4, some compounds a bit like P4, is that actually, as Trine showed, it's actually exposed through different routes. So it's what we call aggregated exposure. So basically, have different routes. So it's actually a bit challenging to do this reverse dosimetry. So what we suggest in this case is actually to do forward dosimetry. So in this case, actually, we start from what is a known exposure. So from the biomonitoring studies, and we actually try to evaluate. Because also in this case, we have plasma concentrations. We try to evaluate between external and the plasma concentrations. And then we actually predict with the PBK model what will be the unbound concentration in in these target tissues, liver and kidney, for example, and then actually compare to the benchmark concentration. So ideally, in the end, we would have something like this. This image from this paper. Basically, you have a distribution for both the effect concentration, but also distribution from the internal exposure concentration. Then we can actually put them together, as Trine said, and try to calculate the probability of a certain hazard or a certain risk. So more specifically, not before, although I already said something. So indeed, this is the case study we selected. I mean, we have more compounds in ONTOX, but this is the one we really try to integrate with all the tools. So it was chosen for different reasons, but indeed it causes liver and kidney damage through different mechanisms. I have to say this is also like some of the work was developed synergetically with the PARC. I'm not sure what is the work package, but it's a work package that develops PBK models. So we had a lot of cross conversations about this because there is quite a bit of information on P4 in literature. There's quite a PBK models in literature. So obviously it doesn't mean all of them are suitable for what we want to do. they vary in species or assumptions. Maybe not all of them are updated to the new data that exists. And so what we know for in general PFAS is that generally not well predicted with the common cues as for other compounds. Because we have quite a bit of data in literature, but this data also varies a lot, and I will show a bit of an example. We know they bind very strongly to albumin, which is an issue both for in vivo but also in vitro kinetics if you use your fetal bovine serum, for example. And we also know this compound is mostly ionized at the physiological pH, so it also means it probably has very slow permeability, because usually you assume more neutral compounds like to permeate easily. Ionized compounds permeate less easily. And we also know that the excretion is mostly through the kidney and biliary excretion. There's quite a few transporters related to it, which also makes it a bit challenging. So the idea that we, so the strategy for us for the PBK model was to, sorry, I think teams coming in, was that first we want to characterize data that we need for the PBK model and characterize the uncertainty and variability, and eventually then really proceed to do the IVIV for these parameters, but also calculate the partition plasma coefficients. We'll evaluate the model with rat and human ex vivo data, because there's some data from either post-mortem or biopsies in humans. And eventually actually compare there's human volunteer data to understand how well the model predicts and if we need to fit something. And then based on this, we can actually build our population model and add uncertainty of some parameters to this model. So we actually take into account both the variability in population, but also uncertainty of some of the parameters. And eventually this probabilistic model can actually be compared to the biomonitoring data. So this is just an example, but there's for other input parameters, there's more examples of this. So for example, so diffraction and bound in plasma, so how much the compound binds to plasma and specific to albumin. There's quite a few values in literature, and I collected a few of them. They also usually come reported with either the standard deviation or confidence interval. So I actually wanted to plot some of them in this density plot. So basically you see in the x-axis is the fraction and bound. And you see, you know, some of the methods kind of overlap, obviously. There's this one that is a bit outside of the distribution. So this one I actually excluded, but I put the other methods, the values from the other methods together. And obviously we see there's quite a bit of well, in this case, I don't want to say uncertain. There's quite a bit of confidence interval for this fraction and bound. But because the fraction and bound is measured using human plasma, it will both account for the human variability, but also for uncertainty of the different methods, because different methods might indicate slightly different fractions and bounds. So we wanted to disentangle both of these, because the variability is taken into account by making a population model, but the uncertainty, we need to incorporate it separately. So I'm not going in detail, but it's just to say these are some of the considerations you need to do actually, like how to disentangle, what is variability, what is uncertainty of the method. So we actually can also update. And also if you look at different populations, we can again also update it based on this. So we took the different in vitro methods, did IVIV, also calculated partition coefficients. I'm sorry, I realized this should come as an animation. Now you don't see it. So one thing that I wanted to look at the rat data is because we can actually evaluate some of the assumptions that we had. Also, if I look at the PBK models in literature, so some of the things that I look, for example, if I do a PBK model that is perfusion limited, so this means it's a PBK model that assumes the compound has no problem crossing the membranes of the tissues. I also did some simulations where I actually then considered the permeability into the different tissues as being a bit limiting. And then the third case, so that's going to be the black line. If I actually also put that endothelial, so basically this is the barrier between your blood and the tissues, is actually also permeability limited. And actually this is the one that I see causing the most differences. And actually, when I looked at the tissue concentration of P4 in the rat, I actually saw that by having this endothelial barrier represented is actually the way that I get the most similar partition coefficients for the different tissues. So this is what we accepted that we would incorporate in the model. And what I want to, because this is quite complex, but obviously what I want to summarize here, by doing this test and looking at the rat and looking at the different permeabilities, we can actually conclude that the blood interstitial fluid and the cells are not usually in equilibrium in vivo because of this low permeability. So obviously it just highlights really that we need a PBK model to understand what is actually the concentrations in the target sites in the organs. And finally, we have the one human volunteer study. So someone took before like a very small concentration and measured in plasma and urine and in feces. And so here I wanted to, hope you're seeing the right screen. I wanted to compare the original model. So basically, if I don't actually do all the changes in IVIV, so if I assume just a normal log P based calculation of tissue plasma partition coefficients, if I assume there's a passive renal clearance, so that the renal clearance can be represented just with the normal kidney clearance, and that there's also no permeability corrections for the endothelium, for example. And this is what we get in the blue line. So here also for the human data, you really see that it's under predicting the plasma concentrations and then we over predict the kidney excretion. But once I do these corrections, and I also needed to fit the kidney clearance because the P4 is heavily absorbed in the kidney, we actually see then the predictions are much better. But also we see, because we didn't have liver kidney tissue concentrations are human, obviously, but we actually see then how much it affects when we improve the model. So we can actually also assess a bit the uncertainty of a very basic model to the model that we built here. And so finally, we conclude, okay, we have a human model that is at least predictive of our volunteer data. So what we want to do then is consider the population. And when we build the population, there's different aspects that we set it to vary within the population, but most importantly for P4 is the kidney, is what we call glomerular filtration rate. So that's the rate that the blood is filtered through the kidneys, but also the albumin concentration in individuals, because these obviously will vary between different sexes, will have different distributions, but also for the different age, you're also going to have differences. And so we have a bit of an example, like basically you have to set these distributions in the population, and then you can finally make a simulation of the population. So here we have comparing to the volunteer data that we had. We have the population model, and you can more or less see then where this specific individual actually sits within the population. And so, so this is the point that I am at. So obviously now I want to add the uncertainty of the parameters and to run the simulations for the actual steady state, because the data, the PBK model that I showed was for single exposure, but actually before, because it accumulates so much, it takes more than 5 years to actually reach steady state. And what this means practically, it will be very heavy computation. So this is why the results actually take a while to obtain, because we need to optimize everything that it becomes at least, if you're doing simulations for 3 days, that actually they are the right ones. And eventually, when we have this, we want to compare to the biomonitoring data. We have already some idea that actually we, from pilot evaluations, that we might be over predicting a bit. If we take estimates of exposure, we might predict a bit the plasma concentrations. So it might have to reevaluate in the end, but obviously, exposure estimates are exposure estimates. So in the end, we're also going to perform the QIVIV also using actually what was measured in plasma concentrations in different individuals, because it also exists, and then we can evaluate both strategies. And that's it for my presentation. I don't know if there's doubts or comments, but you're welcome to say them.
Yes, thank you so much, Susana. There is actually one question in the chat by Chris, who says there are many assumptions made in PBPK models that are reflected in the math. Typical assumptions, for instance, include that equilibrium between plasma and tissue is reached before blood leaves the tissue, which I think you mentioned as perfusion limited, and the liberation from albumin is slow compared to that. Otherwise, binding would not be relevant. Could you indicate some of these assumptions and use in the math and how you tested their applicability?
Okay. Thank you, Chris. I mean, it's an interesting question indeed. It is indicated, I think, usually that compounds that have very high binding to albumin also have indeed like a slow resorption. So actually, like it takes time, and most PBK models, and ours is not different in that case. We assume it's instantaneous. What we call partition is just an instantaneous process. And the reason for this is mostly because there is no data. to model that there's a slow desorption from albumin. Obviously, we could argue that this is a bit of a reason why, in this case, the endothelial permeability, if we set it to a certain value, so we make it limiting, that actually might artificially represent a bit this slow desorption. We also know sometimes this is used also, for example, when compounds bind very strongly to fat, for example, they also have a slow desorption from fat. So by making the organ somehow permeability limited also solves partially the problem. That's it. I mean, if you would really want to do this, you would have to measure experimentally actually how long it takes for the compound to desorb albumin. So if the data exists, please tell me. But obviously there's also it's always a bit tricky because then a lot of the data we have still is in vitro, and in vivo obviously going to have even for plasma, you're going to have slight different considerations because maybe you have more lipids in blood that are also going to affect the binding of PFOA. That's it. But another thing is also some of the reasons why PFOA has slow permeability is because it really likes to sit on membranes, phospholipids. So this is also how in the end has slow permeability is because it just kind of likes to stay there. And indeed, that's all reflected in the test that we did for the PBK model.
Yeah, well, I hope that answers the question. There are no more questions, and also it's 4 already, and we still have 2 presentations to go. So I suggest we go to the next one, which is Julen. If you could please share your... Yes, perfect. We're looking at your slides. So, well, the floor is yours.
Great. Thank you very much. I was also thinking that we are running out of a bit of time. So I would go as smooth and quick as possible through my presentation. That is indeed now talking about the in vitro approach for hazard identification and also characterization. And I put that with a highlight because I wanted to actually discuss with the people here for attendees. I'm not sure how many people from the attendees are in vitro guys or girls or whatever. But in any case, I think it would be nice to have an afterwards discussion about it to see what you think about the approach. So the schedule is actually died already for my 15 minutes, so I probably have even less. But I will be really focusing on OPRA that we already did in the 2 presentations before, but in this case applied for toxicology or for hepatotoxicity, and then I will introduce adverse output pathways and then how we use them to develop the in vitro test batteries, and finally I will discuss a bit of a data interpretation that it includes the weight of evidence, benchmark response, benchmark concentration, and potency estimation. These very the typical things that we always address when we want to do hazard characterization, right? But that's the most interesting part for me, at least. Yeah, okay. So I think we saw twice, or at least once, this OPRA decision tree, and we're going to focus, as Trine already said, in the 4 package 7. This is, again, the hepatotoxicity 4 package. And we have 2 case studies within the hepatotoxicity study, that is liver cholestasis and liver steatosis. So the increasing concentration of bile acids in the liver and the increase of concentration of lipids in a very, very general way of describing them. So this part here that we were discussing about, that is the hazard part, first has kind of a hazard identification part. Here it's called hazard probability, but in a way, in the case of the in vitro part, we have 3 levels. So the first one is basically testing for cytotoxicity of your chemicals, so which are basically you want to always work with the concentrations that are not cytotoxic. That's why I call it level 0. It's not really for evidence, it's more for methodological purposes. And then you go to level 1 and level 2. So for level 1 would be more transcriptional assessment via PCR, omics, you name it. And level 2, a functional assessment. And afterwards, once you have the data, that it would be a bit afterwards in the 8th slide, I guess, And it's when we can discuss about the intensity of modulation that is happening in your data, the weight of evidence. So basically, which of the parameters that you're measuring are more important than the others, or the benchmark concentration and the benchmark response. And you can calculate it from there. And how, or at least the approach that I'm trying to explain here, how can calculate these kind of parameters. Right? A bit slow. Sorry for that. Or maybe it's just me. But before we go into the data itself, I wanted to say that we developed some in vitro test batteries that are composed of these two levels, so the transcriptional one and the functional one. And these are mechanistically anchored in adverse outcome pathways. I don't know. I'm pretty sure that you, almost everyone that is here listening is very aware of what adverse outcome pathways are, but basically it's just understanding or a representation of the mechanistic data from the initiating events that are called molecular initiating events at the very, very, very cell level. And then it goes, how it proceeds, how it goes through other key events until the adverse outcome. And I already talked about that our case studies, or in this case, adverse outcomes, are liver steatosis and liver cholestasis that are here mentioned. So you can find the AOP networks that we have developed within ONTOX that are published now. So if you are interested in that, feel free to go to these two publications. And what it does, as it is a mechanistic evidence, it supports which assays or which key events you're going to tackle if you want to address the pertinent adverse outcome, right? So in this case, for example, one of the novelties that we introduced in these AOPs is introducing the evidence that there is to support the key events and the key event relationships. As you can see, for example, here, some of them are bigger than others. For example, here in steatosis, intracellular lipid accumulation is more relevant. Basically, it's the definition of steatosis of one of them. So it got a bigger box, if you may. So it was more talked about or more discussed. It has more relevance. And in the case, for example, here, fatty acid synthesis, fatty acid uptake, etc. And these relationships here between these three key events that I'm mentioning now are, especially in steatosis, relevant. So knowing that, we proceeded to develop the in vitro test batteries. And this is basically what we have been doing the past years. In the case of liver cholestasis, we have, as I already mentioned, a level 1 and a level 2 for both cases. The first one is the transcriptional fingerprint. The second, different key events targeted by different assays. I will focus now more into the liver steatosis because the data I'm going to show is related to that. So, especially relevant now is the intrahepatic lipid accumulation and the fatty acid uptake. So just as a summary, what we do with the in vitro test battery, or what they are, is just different mechanistic assays that are covering different key events or key biological pathways we have here to identify the hazards and understand the mechanism of action. And just to mention that they are in process to be standardized because we have some TOX templates and SOPs already in place. So I think that with the next slide, no, sorry, not yet, because I wanted to also do a bit of a, that you see that these in vitro batteries have been already introduced as 2 papers, one for liver steatogenic potential of chemicals and the other one for chemical induced cholestasis, and they are out there. So if you're interested in this, again, go to it because they're very, very interesting articles. But there are 2 others that are ongoing now, and I'm going to use the data from one of them. So if you see some kind of coding in the chemicals that have been used, well, it's because of confidentiality purposes. That said, and I think I'm in time, if you're not understanding anything until this point, now is the moment to come in. So please come back. And because it's just about data interpretation and about how we're going to proceed from hazard identification to hazard characterization. And I'm trying to make make it a bit simple. So instead of different levels and so on, I will focus on only level 2, which is again the functional assessment of 2 key events that are relevant for steatosis. That is intrahepatic lipid accumulation and fatty acid. So these are the results that you get from in vitro setting, right? A different one, a PC, that stands for positive control or a priori positive control, right? 1, 2, 3 until 7. That's what we tested. And then 2 negative compounds. So what you observe is that there is an increase in lipid accumulation, the higher the concentration, right? In almost all the positive compounds, both for lipid accumulation and fatty acid uptake. And in a very simplistic way of interpreting this data, you would get, okay, if I have a positive compound, if I have a positive modulation of, or an increase in modulation of one of the concentrations that I'm testing for per chemical, then it gets a 1, it's a positive. So you will do a statistical assessment. You would say, okay, positive compound 1 gets positive when it's in a higher concentration for lipid accumulation and fatty acid uptake, right? It's translated like this. So you get a very binomial, very categorical system, which doesn't really give much more information if you want to proceed to other aspects of hazard identification. So after the concentration response modeling, after the statistical analysis, normally with an ANOVA, not an expert in that, but that's commonly what is done. Maybe you could derive a kind of a BMC, because you can say, hey, the lowest concentration that is able to activate one of the key events, that is my BMC. That is my benchmark concentration. But I think we can do a bit better, right? How can we do it better? Well, first of all, with weight of evidence. And what it says weight of evidence is which of these key events that you are testing is more relevant towards basically that it appears or that it predicts the adverse outcome. So in this case, what we did was using the data gathered by the generation of the AOPs and use the Bradford-Hill criteria that has 3 different parts: the biological plausibility, the empirical evidence, and the essentiality. What did we do? Okay, biological plausibility is related to the causal association, meaning is there any evidence out there in the bibliography that links that the activation, and I will make the same example as I was doing, that fatty acid uptake is linked to steatosis. Well, the higher the link, the higher biological plausibility. Empirical evidence, what it does is, instead of just the link, is there any dose response or time concordance with these 2 different key events? So fatty acid uptake and steatosis. And essentiality would do is, if I test with my assay fatty acid uptake, to what extent am I able to predict the adverse outcome for steatosis? So with these 3, you can join them together, and instead of, still will be categorical, but it's still, but instead of 1 and 1, so sorry, 1 and 0 values, you would get an arbitrary unit that would compare between both. So in this case, lipid accumulation is 2.1 and fatty acid uptake is 2.3, meaning that fatty acid uptake somehow is more linked in terms of evidence towards steatosis than lipid accumulation itself. After this, we have these results. What you could do is, okay, let's try to get some benchmark response to get measure of intensity of your response, because PC1, I don't know if you, I can show you actually. I was going to say, I don't know if you remember, but the answer, the modulation that you get from PC5 and the modulation that you get from PC3 is not the same. Then you can somehow see that the adverse outcome would be more severe in the case of PC5. Now it's a lot too theoretical in a way, but I think it's it's proper that we start talking about these kind of things, especially in vitro models. So what we did, and I will go fast, and sorry for the time, is that with an app that it was developed within Work Package 9 in ONTOX Stats, we changed from statistical significance to biological relevance by, instead of doing a simple ANOVA, we see the variation. So we calculate the benchmark response based on the lowest concentration that did not exert any modulation. So it's what I'm showing here. So all of these concentrations together have a different modulation, obviously per key event, and that it's compared to the rest of them. If the concentration of the chemical falls within the benchmark response, then it will be a negative. If it falls out, it will be a positive. So what you can do also is take that benchmark response, let's call it 10, so it will be around here, so 1.1, and calculate how much of a difference is between, or the relative difference between the highest induced modulation compared to the benchmark response one. And with that, you will never get any more a qualitative assessment, but a quantitative assessment, right? Because you don't get any more the 0 and the 2.1, you would get 0, 7.77, 1.2, etc., etc., etc. And then to finish, I showed you only 2 key events, but there are many more. And what you can do after that is you bind or you sum them together, and you get this. Simple, very simple. A lot of work to get to a simple, straightforward graph here that it shows basically the statogenic. So the PCE, that we said, the positive controls have a higher statogenic score than the non-statogenic. That can be used for categorization, prioritization, but what about hazard characterization? What can we do with this? Well, why not establishing a cutoff there, for example, that it would be 6, in this case, 6 of a statogenic score. And with that, and as an example, we're going to take PC1 and PN2, so just a random positive control and a random negative control. Actually, I see that PN is not negative control, so there's a typo there. But in any case, PC1, you can cut when the exposure of PC1 crosses with the cutoff that we have established, that is 6, because 6 is the difference between being statogenic and non-being statogenic. And in this case, what you can do is somehow refer to a benchmark concentration that is 2,900 micromolar for PC1. What happens for a negative control? That it never crosses it. So you'll never be able to get that benchmark concentration, at least with the concentration that you have been testing. After this, and this is my last slide, I promise, you can do a potency approximation, something that Susana already mentioned, actually. Once you have the nominal BMC of that you calculate in vitro, you can do in vitro distribution models to check for the effective concentration, or basically the concentration that is within intracellular, so the one that is affecting the key event itself. And after that, you can do either reversal symmetry, something that I'm not aware. I mean, I don't know how you guys do it, but apparently you can do it. And then you can go to human relevant concentrations, and that would make you a potency approximation. And just a curiosity to say that the Cmax of this PC1 in humans is around 600 micromolar. So with this a priori proof of concept simplistic approximation, we can already try and play around with this approach for potency approximation. Thank you very much. And sorry for being more than 15 minutes.
It's okay. Thank you so much for the nice presentation. There are no questions yet in the chat, and I think for the sake of time, we will not wait for any questions. So I think if people have any questions still, then they can email them to you, I think, right?
Yes, please. For sure.
Perfect.
Happy to discuss anything.
I'll put them in the
Great.
chat now if they want to. And Julen, I think you're okay to monitor and answer if...
Definitely.
Perfect. And we are now going to the last presenter, which is Fabian from the VHP4Safety project. And of course, well, it's only 10 minutes left in the original, meeting as we scheduled it. But yeah, I think we will go a bit over time. I hope that's also okay with you, Fabian. I don't know if you have any appointments waiting for you after this. You're still on mute.
No, I'm fine.
Okay, perfect. Thank you so much for your flexibility. If people have to leave at 4:30, then of course the recording will still be published online. So if people then want to rewatch this part, then that is also possible. But of course, we would also like for you to stick around also because then you will have the opportunity to ask any questions about risk assessment in VHP4Safety at the end of the presentation. So Fabian, the floor is yours.
Right, you can see my screen now, right?
Yes.
Okay, great. So I'm Fabian. I'm a postdoc working at the VHP4Safety project, and I was asked to present here the case study that we're using to show how we use NAMs to do a chemical safety assessment for a compound silicristin on thyroid hormone-mediated developmental neurotoxicity. So a little bit of background into the case study. Thyroid hormone is a key regulator of neurodevelopment, where an inadequate amount of thyroid hormones can lead to either an adverse effect on cognitive functions or motor functions. And currently, all of these thyroid hormone things are assessed in animal models only. And we know that these animal models are not sufficiently capturing the effects of thyroid hormone-mediated effects. This is especially true for thyroid hormone transmembrane transporter called MCT8, where in patients that have a deficiency in this MCT8, we see that they have decreased thyroid hormone in the brain and resulting severe neurodevelopment effects, as I mentioned earlier, the reduced cognitive function or reduced motor function. And what's curious is if we have rodent models where we've knocked out the MCT8, then we don't see any of the neurological effects. So we know that we cannot assess effects on this MCT8 using traditional animal models. So this is really the perfect case study for seeing how we can use NAMs in this approach. So in this case study, we focus mostly on silicristin, which is widely used as an MCT8 specific inhibitor, but is also a compound that is commonly found in dietary herbal supplements in a mixture that's in these supplements. So it's also widely consumed by the public. So really, this is a combination of this is really interesting to see, okay, what can we do with NAMs to answer this research question? So we set out to see if we can answer the question, does silicristin influence the thyroid hormone-mediated brain development in the fetus? And here you can see an overview of the process flow, as we call it in the VHP4Safety project, to see how we approach this risk assessment. So we first start with estimating a realistic exposure, and then we use kinetics, PBK modeling to get to an internal exposure estimation. We then use a variety of in silico tools and in vitro tools to characterize the biological activity and also to identify any sort of point of departure. And we mainly do that based on developed AOP, and we use the AOP to identify key targets for which we make in vitro assays, and then we feed this into a QAOP, answer this question, and at the end we compare, or the idea is to compare the risk assessment that we did before this to already available in vivo data from human clinical data and also a zebrafish study. So how did we do the exposure scenario? Well, as I mentioned before, silicristin is part of a mixture called silimarin. This silimarin is part of a herbal extract that's usually sold as like a liver detox or a breastfeeding supplement. So the main exposure route that you are interested in is oral exposure to ingestion of these products. And to find out how much people are exposed to this product, we looked at dose recommendations that you can find on information from product leaflets. We did this for both the Dutch market, but also from a big American dietary supplemental label database. And we simply look at, okay, what is the amount of silimarin that these product leaflets state they have, and what is the serving size that they also state for that. And based on this, we derive some RDIs for silimarin itself, and then we use literature search, and also we are currently measuring the silimarin abstracts to see how much of this silimarin contains, how much silicristin is in this silimarin mixture, all to derive a sort of recommended daily intake for silicristin itself. This recommended daily intake for silicristin we can then put into a PBK model that we are currently developing. It's a flavonolignin PBK model, and here we can use the RDIs to derive both a maternal systemic concentration, but also a fetal brain Cmax concentration. And we also test silicristin in a intestine ex vivo intestinal model to look for the gut absorption, as we've noticed from the literature that this is a very critical parameter. And we can make a sort of uncertainty estimate for this to see if our PBK model is overexposing or underexposing, overestimating or underestimating the predicted concentrations that we see. We then developed an AOP specific for thyroid hormone mediated developmental neurotoxicity on neural cells. You can see the AOP here. It's published already, so it's available. And we use this AOP to really define which molecular initiating events are important to reach an adverse effect for cognition, and also to identify which key events are important in this whole sequence. And this was used to inform the selection of which in vitro assays do we need to use, which in silico prediction tools do we need to use. So based on the AOP, based on the AOP, we found that we mainly have effects on thyroid hormone receptor activation, dehydrogenases, and thyroid hormone transport. So we looked for freely publicly available in silico tools. Some of them have also been developed in the VHP4Safety itself, but some of them are external tools. For example, we have the OECD QD box, which was developed in VHP4Safety, which is an AI agent that that assesses the OECD QSAR toolbox. We also use QSAR prediction tools that were developed, and also some docking tools. And externally, we also have some QSAR tools and docking tools that are relevant for the molecular initiating events that we are interested in. So you can see here on the right all the predictions from these different tools. And you can see that for mostly we don't see an effect on thyroid hormone receptor activation, but we do see a predicted effect for MCT8, obviously, and also some from dehydrogenase inhibition. So we then use the selection of in vitro assays in different cell models that are representative of neural cells, so astrocytes, neurons, and oligodendrocytes. And we checked in each of these cells, okay, what is the effect on thyroid hormone transport? What is the effect on dehydrogenase? What is the effect on thyroid hormone receptor activation? And we found that there was not really an effect of silicristin on deiodinase activity, but we do see an effect on thyroid hormone transport activation and also on thyroid hormone receptor activation. However, we think that the effect on thyroid hormone receptor activation is mediated as a sort of downstream key event because of the decrease in transport instead of an actual interaction with the thyroid hormone receptors. And this was based on the in silico predictions that we did before this. So following this, we looked at simple cell models that are overexpressing some of these thyroid hormone transporters, and we tested silicristin in multiple concentrations to derive a benchmark response and also a benchmark concentration then for each of these transporters. And we also looked further into what is the effect of silicristin on further developed key events. We used a more sophisticated neuronal astrocyte co-culture cell model for this. And here we also look at the effects of, for example, KLF9 expression, BDNF expression, both on a genetic scale and on a protein level. And also look at the network functioning of these neuronal cells using multiple multi-electro arrays. And you can see here on the right already that if you do that, and we did this both for silicristin, but also for varying concentrations of thyroid hormone itself. So that is independent of the chemical actually, which is also how it is in your adverse outcome, of course, because the effect is propagated by a decrease of thyroid hormone and not necessarily through a binding of silicristin to any of these thyroid hormone receptors or whatever. So you can see that the effect is very prominent on KLF9, but we don't see a clear effect on BDNF yet. And all of this data that we are curating for all of these different key events and molecular initiating events are collected and are being built or are used to develop a quantitative AOP. This is currently ongoing, so I can only I showed you here the cisplatin QAOP that was developed already. That's a sort of example, but the idea is that using this QAOP, we can predict the effects of silicristin on all these downstream key events. So you can see here, for example, for cisplatin, DNA damage would be a molecular engineering event. You can predict how this relates to kidney cell necrosis and then to kidney failure. And something, a similar approach is what we are planning to use for the thyroid hormone mediated developmental neurotoxicity. So to wrap it up, we made a thyroid hormone developmental neurotoxicity risk assessment based on relating internal exposure to a QAOP. So it's mainly based on an AOP approach. And we use various data streams, both from within the VHP4Safety project, but also from external sources. Currently, the PBK modeling and some of the in vitro data generation and development of the QAOP are still ongoing. So I can't show you too much about it, but I hope at least the outline of everything was clear. And in the end, once we have all this data curated and all the QAOP is done, we can make a comparison also to human clinical data from MCT8 deficient patients and also from a zebrafish study where we exposed zebrafish to silicristin. So thank you all for listening, and also thank you everyone who worked really hard on this, especially Natalie, Ellen, Lianne, Rob, and Nienke, who were really the thinkers of the founders of this risk assessment.
Yes, thank you very much, Fabian, for your presentation and for doing it so quickly, because we're only 4 minutes late. There are no questions in the chat yet. I don't know if anybody would still like to ask a question, or maybe about the previous presentation, because I think we still have a bit of time. Well, officially we're 4 minutes over time, but still quite some people here.
If you have a burning question, you can also raise your hand instead of writing it. That might
Yes.
Easier. And wait for a minute. All right. Well, I think we can let everyone go on vacation then.
Yes. Okay. Yeah, thank you all very much for attending the webinar and the presenters for their presentation. This was the last webinar of the ONTOX-VHP4Safety webinar series. And we would also like to thank AlterTalks for their support in organizing this webinar series. Yeah, I think then this brings us to the end of the series, right? So do you have anything to add, Marie?
No, I mean, yeah, thanks for everyone that was involved in the series. I think it was a long work this year to make this series happen. The whole series, you can rewatch it on YouTube. If you have any comments, any questions, don't hesitate to reach out to us. Feel free to register to the ASPIS Academy LinkedIn group. We always post updates about what we're doing there.
Yes.
And in the meantime, have a happy holidays.
Yes, happy holidays and a good 2026.
All right.
Happy holidays. Thank you.